All right, so be it. Endospores. So what are they? They have nothing to do with the fungal spores reproduction that you can see when you kick the mushroom and spores just fly out as a cloud. Endospores are special structures in the bacterial sporulation. You can see the idea of sporulation right on this illustration. It starts with the formation of so-called forespore, when a special membrane is formed around an additional copy of the bacterial DNA. The cell carries 2 copies of its genome. Genome is surrounded by the membrane. Membrane then is surrounded by cortex. Cortex is surrounded by additional protective cover. What you have to understand is that during sporulation, the only part of the bacterial cell that gets protected is DNA. Does that make sense? Cell doesn't care about cytoplasm, ribosomes, anything else. Only DNA. And then spore is released. It's not released just like released. Imagine like in Star Trek, when USS Enterprise explodes, Captain still can save himself in like a capsule, a pod, like, you know, the pod. Okay, that makes sense? That's the survival pod for bacteria. Cell itself, right here, cell dies, but DNA persists. And if you will go into the dry environments, and in this room, the most dry environments would be tops of the cabinets, anything that has a lot of dust on it. That's where you're going to find a ton of endospores. And if you collect it well and you put it in the agar, those endospores that can sit there for days, months, years, will start to germinate. Germination is the process of endospore turning into the vegetative cell. Vegetative means growing cell. 2 major stimuli for germination is the presence of water and presence of certain nutrient, a germinating factor. Usually when endospore starts to germinate, nutrients are abundant, like on the agar plate. Among the endospore forming bacteria are bacilli and clostridia. You know the Clostridium genus pretty well from your experience, Clostridium tetanus, bacteria that causes tetanus. It's usually acquired when you do things like stepping on the rusty nail. It's in the soil. The endospores are in the soil. And when those endospores get into your muscles, into your tissues, plenty of fluids, plenty of nutrients, they start to germinate. Does that make sense? Same goes for Clostridium botulinum, causative agent of botulism. When you don't preserve food properly, endospores can persist there. Okay? You consume, Clostridia starts to grow, you have botulism. Bacillus anthracis, the causative agent of anthrax, is one of the most, probably one of the most feared bacterial infections. You all remember anthrax attacks in 2001, I suppose. There was a lot of fear about that. Because now all male, and it's not a joke, it's not hyperbole here, all male in the United States is checked for anthrax. There are special machines, PCR machines, that check every piece of mail for the presence of anthrax endospores. And anthrax can survive for many, many years. In the soil, it's an infection of cattle, and if you will bury the infected cows, anthrax will go in the form of endospores in the soil and will survive there for years. So if eventually somebody dips into that cemetery, cattle burial place, the person can acquire anthrax. And it happens from time to time. Endospores are extremely hard to kill. You cannot kill them with boiling, for example. You can kill them with autoclave, you can kill them with gamma radiation, but not with boiling. Okay, so you got to remember that. So this is the electron micrograph of the endospore, which doesn't look like a normal bacterial cell. And these are some microbes, shape-wise it's bacilli. You can see the bluish endospores inside of the pink stained cells. Make sense? Endospores protect genetic information from harsh conditions. Yes?
Didn't you say that the cells die?
Oh yeah, yeah, cells die. So these guys, these red guys, they're about to die.
Oh, okay.
Yeah, they're gonna die.
Okay.
So endospore formation is stimulated by worsening conditions. Does that make sense? Yeah, they're gonna die.
One more quick
Yeah, sure, as many as you want.
So once you put the endospores on like a
The agar.
agar,
Okay.
right? With the presence of water and nutrients,
Yes.
how does it start to replicate its DNA and
It's a great question. The short answer, we don't really know. People study the signals that regulate germination. We don't know like the detailed mechanism. There are probably some protein-mediated sensors, so proteins in the cortex that recognize those nutrients and initiate the disassembly and replication of DNA. But how exactly it happens, I cannot tell you. So it's still, this whole process, we're still studying. Make sense? Good. It's a great question, actually. Cell wall. Now, we talked about the cytoplasm, we talked about the nucleoid, we talked about the ribosomes, and cell membrane. So we covered 4 structures that are present in each and every bacterial cell. Now we're going to talk about the cell wall. Surprisingly, this is the part of the cell that's not in every bacterium. There are microbes that simply do not have a cell wall. All they have is a membrane. But let's talk first about the ones that do have. You've heard the terms gram-positive and gram-negatives. Okay? We distinguish them based on specific staining. Turns out that these 2 types of microbes, gram-positive and gram-negative, are fundamentally different in terms of the cell wall structure. Bacterial cell wall consists of the polymer called peptidoglycan. Based on the name, peptido means it consists of what elementary building blocks? Peptides consist of, but peptides consist of amino acids. When we say glycan, what is the synonym? Broader. Carbohydrates, yes, carbohydrates. So we have amino acids and monosaccharides, right? It turns out that long chains of monosaccharides, N-acetylglucosamine and N-acetylmuramic acid, are present in both gram-negative and gram-positive microbes. These long chains of carbohydrates are linked by the peptide linkages. You see, these peptide linkages. So if you look at this structure, you'll notice that the structure is a lattice. If you would look at the fabric of your clothing, you will see that it also consists of multiple threads that are perpendicular to each other. A lattice. A lattice actually is a pretty strong structure, you know. So this cell wall is quite mechanically resilient. Does that make sense? There are minor differences in terms of the length of peptide links between gram-positive and gram-negative, but overall, both of them have chains of monosaccharides connected by the short peptide fragments. Okay. Now there are, we're going to talk about the implications of that structure in a second. Now I want to talk about the fundamental differences between gram-positive and gram-negative cells. Gram-positive cells have very thick layer of peptidoglycan. Underneath, they have cell membrane. So it's like a sandwich where you have a bread and whatever meats you have on top of it, and that's it. Okay, it's not two pieces of bread, just one. Bread and then meats. Membrane and peptidoglycan. Does that make sense? Huh? Open face? Well, thank you. I learn some new English terms every day. So, open face sandwich, all right? The layer of peptidoglycan is penetrated by the molecules of teichoic acid. Long hydrocarbons, pretty much, that serve multiple purposes, including adhesion of the cell to the surfaces. They may serve as an antigen for immune recognition and also assist in transport of molecules across the cell wall. Make sense? Teichoic acid is unique to gram-positive cells. Gram-negative cells are the classic sandwich. You have cytoplasmic membrane here. Then you have fairly thin layer of the peptidoglycan, cell wall itself. And on the outside, you have another membrane that is called outer membrane. So you have membrane, cell wall, membrane. Okay? Now the outer membrane has many structures that are, again, unique for gram-negative microbes. Some of them are the porine proteins. Porine proteins are essentially the channels that span through the membrane and allow transport of various molecules from the outside of the cell into the cell and vice versa. Does that make sense? Like doors. The membrane is attached to the cell wall by the murine proteins. But probably the clinically most important molecule that can be found in the outer membrane of gram-negative microbes is lipopolysaccharide. So it's marked here as this red sticks on top of the membrane. Lipopolysaccharide is also called endotoxin. Well, I hope I don't need to explain the meaning of the word toxin. The part endo, what does that mean? Usually when you say endo, inside, yes. So it's a toxin that comes from the inside of the cell, that is a part of the cell. Every gram-negative microbe has endotoxins. Does that make sense to you? Every one of them. Interesting feature of these endotoxins is that they don't produce immune response in the sense of antibody formation. So it, you're not going to have a long-lasting immunity against gram-negative microbe. Does that make sense? You get infection with E. coli once, you get infection with E. coli twice, and the toxin will still cause all the symptoms. Now, what are the symptoms? When you get sick with gram-negative microbe, it's a fever, very high fever. It may cause immune response that is blown out of proportion, and it causes the toxic shock and may lead to hemorrhage. What is the structure of that lipoprotein? So it has the O antigen, the core, and the lipid fragment. Or antigen and the core, both carbohydrates. Lipid fragment, lipid A is obviously lipid. Okay. Now, we do not produce antibodies to it. Which implications does it have for us? Imagine that you have salt and water, and you want to make isotonic solution for IV drips. It should be sterile, right? It must be. So mix salt with water in the desired proportions, put it in the bottle, stick the bottle in an autoclave and run it in the autoclave for, 40 minutes. Is it sterile? I bet you. It's as sterile as can be. Then you have a patient, you put that IV drip in the patient, and in, 30 minutes or an hour, patient develops skyrocketing fever. There's no infection, just the fever. And the toxin, lipopolysaccharide, is known to be pyrogen, which means a molecule that causes fever. Does that make sense? So even if you take bacterial cell, gram-negative cell, destroy it, lipopolysaccharide will still be present in the solution. So technically speaking, your solution will be sterile, absolutely sterile. But it does not mean that it's not containing pyrogens. You see the difference? It may still contain fragments of the dead microbes. Question?
Okay, so you're saying that the bacteria is dead, but the LPS still exists.
LPS still exists and causes fever. And it's a big problem because all injectable materials have to be checked for pyrogenicity. The classic test, why I did that, that's why I know it. Classical test for pyrogenicity is to take a rabbit, inject, according to a certain procedure, the drug, IV. Rabbits are notorious animals, and injecting something IV into the rabbit requires courage and usually two people. Well, if you do it by yourself, you're running a risk to kill the poor fella because you have to hold it down. I personally knew a very nice lady, like this tall lady, about 65 years old. She told me about her first experience with rabbits. She said, I'm keeping him, he's moving. I'm keeping him, he's moving. I'm keeping him, he's not moving. I'm injecting, he's dead. She just broke his neck. So they really active and aggressive animals. And then once you injected it, you stick the thermometer up the rabbit's ass and measure the temperature, which is unpleasant for a rabbit, but other than that, it's okay. There are actually systems that allow you to monitor like 25 cages with rabbits, so keep them restrained. You have those thermometers, and it's all recorded in the computer, very fancy systems. Yes?
Horseshoe crab.
I was about to tell you that.
Oh, okay.
So horseshoe crabs, you've all seen them, right? You've seen horseshoe crabs. No? Well, pretty neat, little critters. And they don't look like a normal crab. They just different. I'll pop up the picture during the break. Anyway, they don't have blood. They have hemolymph, a special liquid. It's kind of a blood, but not blood. And hemolymph contains the blood cells, well, hemolymph cells that are called amoebocytes. Amoebocytes serve a lot of purposes. They transfer oxygen. They also provide immune response. An immune response in horseshoe crab is interesting. When horseshoe crabs are exposed, the bloodstream is exposed to the gram-negative bacteria, amoebocytes form clot right away. They just clot the infected area and prevent the spread of microbe. So a company called Charles River Endosafe started to make a lysate, a prep, special, wouldn't call it a diagnostic kit, using amoebocytes from horseshoe crabs. Pretty much when you mix this reagent with your sample, if there is endotoxin, if there is LPS, which is characteristic for gram-negatives, right? You have a clot in the tube. It's extremely sensitive, like unbelievably sensitive. The detection limit is orders of magnitude lower than the threshold that will cause the temperature, the fever. Okay, it's a fun analysis, but you have to be really, really careful in handling. So you have to do everything fairly accurate to avoid contamination. But other than that, it's pretty straightforward. The only question remains is how do you get hemolymph? To kill the horseshoe crab? Not necessarily, no. They migrate every year, and they crawl at the shores of one state in the U.S. Well, probably two, but one is the most famous. Like crabs, Massachusetts. Turns out Massachusetts, not Maine, surprisingly. Massachusetts is the biggest place where they migrate. And people just collect crabs, bleed them, not completely, they drain some certain amount, and then release them back. And it works. It's really a diagnostic tool now. Very popular. So it's important to detect LPS in the samples. Now, why we pay so much attention to gram-positives versus gram-negatives. Why we pay so much attention to the staining. Well, you can see that structures are very, very different, right? Well, it turns out that drugs like penicillin, vancomycin, well, penicillin and its derivatives, vancomycin and its derivatives, they interfere with the synthesis of the cell wall. They interfere with making those linkages. So instead of a lattice, you have just lines of polysaccharides. How would it change the properties of the cell wall, mechanical properties? Huh? It will be mushy, yes, it will be weak. So what will happen to the cell, bacterial cell, if cell wall is weak and mushy, huh? It will just explode, right? It will lyse. Which microbes are more sensitive? What do you think? Gram-negative or gram-positive? Gram-positive. Why? It's the right answer. The outer membrane, yes, because it doesn't have the outer membrane. So that's why it's important when you, that's why staining is important. If you know at least the type of the microbe in the clinical sample, if you know that patient has a gram-positive infection, you know penicillin, vancomycin, well, they will be effective, okay? If you have a patient with a gram-negative infection, you probably should think about something else, some other drug. Penicillin and vancomycin will not be as effective. Does that make sense? Now, are there any other microbes with other structure of the cell wall? The answer is yes. You've all heard about tuberculosis. So Mycobacteriaceae is the special sort of a subtype of gram-positives. We usually put them in a separate category, the category of acid-fast bacteria. These formerly gram-positive microbes that have plasma membrane and a thick wall made of peptidoglycan have an additional layer of waxy mycolic acid on the outside. This waxy layer of mycolic acid protects mycobacteria from immune response, from macrophages. Okay? Macrophages cannot, well, they can swallow, but they can't digest. Make sense? Turns out mycobacteria love to replicate inside of the macrophages and live inside of the macrophages. Right? You see, so they, instead of, they are living in the cells that are supposed to kill them. Now, we call them acid-fast because to identify mycobacteria we use acid-fast staining. You can see the red acid-fast bacteria in this micrograph. Three important species that I will mention in relation to mycobacterial infections. Of course, you know Mycobacterium tuberculosis, the causative agent of tuberculosis in humans. There is another one called Mycobacterium bovis, an actually group of Mycobacterium avium. They don't cause any disease in healthy patients, but in immunosuppressed, like HIV patients, AIDS patients, they can cause disease that is very similar to tuberculosis. And also Hansen's disease. What is another name for Hansen's disease? Anyone knows? It was killing and debilitating people in Hawaii in the beginning of 20th century. Leprosy. Yes, Mycobacterium leprae is another example of acid-fast stained bacteria. The thing about these guys, they're fairly hard to kill. But it turns out drugs like penicillin, well, not anymore because they developed resistance, but vancomycin, isoniazid, they work pretty well against this formerly gram-positive. Formally. Does that make sense? So you can say Mycobacterium technically is gram-positive, but we put it in a separate category because we use different staining to identify them. Right? Some microbes do not have a cell wall at all. Mycoplasma, the microorganism that the genus that causes different infections, for example, pneumonia and infections of the urinary tract, belongs to the group of microbes that are called millicutes. Millicutes means without cell wall. Microbes with the cell wall are called firmicutes. Mycoplasma is the smallest independently living cell. Its genome is about 530 genes, which is very, very small, like really small. It can be cultivated on special agar. It usually, though, resides inside of the cell. It's so small that regular light microscopy doesn't detect it. You have to do special staining. You can use fluorescent microscopy to see it. But regular doesn't let you see it. Now, what about archaea? Well, they do have cell wall, but this cell wall does not consist of peptidoglycan. They have other types of polymers, although similar. Glycoproteins. Well, what would be the difference between peptidoglycans and glycoproteins? Seemingly, they consist of the same stuff. When we say peptidoglycan, which molecule takes more like space? Carbohydrate, because it's glycan. When we say glycoprotein, it's mostly protein, mostly amino acids with some carbohydrates, right? In some cases, they don't even have the amino acids. They just have entirely polysaccharide cell wall. Before we go next to the next slide, any questions?
These gram-negative microbes are like really nasty.
Yes. If you would look,
Soft and positives.
if you would look at the microbes that cause the human disease, that typical human pathogens, you would probably find Staphylococcus and Streptococcus among gram positives. But then you open gram negatives, and you have a garden variety. E. coli, Salmonella, Campylobacter, Klebsiella, Shigella, all intestinal microbes, gram negatives. A bunch of microbes causing urinary tract infection. Microbes that cause, meningitis, they're 2 gram negatives, 1 gram positive. So Haemophilus influenza, the Hib vaccine that you get, meningococcal infection, Neisseria meningitis, gram negative. Gram negatives are really hard to kill with the drugs. They have also different resistance to cleaning agents. Some of them are more resistant, some of them are less resistant. Okay, but generally, if you look at when we're going to talk about the pathogenic, we're going to go through gram positives very quickly, and then we're going to have like this list of gram negatives that cause different diseases. Going out of the cell wall, there is a covering that some microbes have called glycocalyx. So it's a layer of carbohydrates. And this layer of carbohydrates in bacterial cells usually comes in 2 flavors: slime layer and capsule. Capsule is more rigid. It protects the cell from phagocytosis. Its main function is to protect the cell from phagocytosis. It also increases the attachment of the cell, improves attachment of the cell to the surfaces. Slime layer plays less role in a protection against immune system, but increases the attachment as well. If you would look at some colonies that you have, some of the colonies have what we call mucoid appearance, like mucus, like covered with mucus. It's usually a sign that the microbe that forms this colony has either a capsule or a slime layer. Given what I told you, that capsule and slime layer prevents phagocytosis and increases attachment, how would you call this? Okay, microbe that has capsule versus microbe that does not have capsule. Which one, in your opinion, will be more pathogenic? Huh? With a capsule, yes. Right? So capsule or slime layer is called a virulence factor. Make sense? Attachment. Can increased attachment be a virulence factor? How? I mean, it sticks better to the cells, right? If it sticks better to, I don't know, surface of the intestines, surface of the lung epithelium, it will more likely cause the disease. Does that make sense? So anything that increases the ability to cause the disease is called a virulence factor, right? Now, why attachment is important for the disease? Absolutely. And we try to focus on the clinical aspects. And another clinical aspect in terms of attachment is formation of biofilms. When you walk into the pond or in the river and you step on the rock, it's covered in a slime. That's a biofilm. Why? So microbes can exist in two forms, pretty much, a biofilm form and planktonic form. Planktonic is, you've seen the liquid medium that I used to, liquid culture that I used to inoculate the table. That's planktonic, okay? Biofilm is when microbes grow on the surface. You may say, look, you inoculated the surface, you sprayed it with microquat, they all were gone. Well, it's true. Biofilms are not simply the layer of microbes covering the surface. It's a community. Usually these communities consist of many species that exist in mutualistic or synergistic relationships. Can anyone define mutualistic or and synergistic? Yes.
They help each other.
They help each other. Is that going to be mutualistic or synergistic? Okay, synergistic, they help each other. Mutualistic. Why I use two different terms?
Some are benefiting and some are neutral.
Actually, in mutualistic, all benefit. We usually use mutualistic when we refer to a community where individual members cannot survive by themselves. Synergistic is when they can, but when they together, they're much better off. So they help each other. Does that make sense? So what happens in why biofilms? Think about this. It's not like a bunch of microbes come together at the dinner table and say, why don't we form a biofilm? It's going to be fun. It's a result of evolution, okay? Microbes that can form biofilm will survive with higher likelihood. The reason for this, biofilms protect them from water loss. Biofilms can accumulate water. Biofilms can increase the efficiency of nutrient utilization. Something like this microbe uses glucose and produces butyric acid, and next microbe uses butyric acid and produces something else. Does that make sense? One microbe produces chemical that protects microbe number 2 from microbe number 3, okay, and reduces competition. And we already talked about quorum sensing. All those microbes produce chemicals that regulate the numbers of microbial cells in the community. You may say, well, okay, slime on the rocks, that's very exciting, that's very interesting. How the hell is that related to clinical? If you worked in orthopedic surgeon office, or you have relatives that have hip or knee replacements, you know that many patients who have prosthetic joints have to be on the antibiotics for the rest of their lives. The reason for that is that surface of the joint can be contaminated with microbes. It's really hard to avoid contamination. As much as you try to clean it, it's hard. And if you stick some of those microbes into the joint, into the actual human body, they will start to reproduce. And they will form biofilm. And taking antibiotics will restrict the reproduction. It will never clear the microbes, but it will at least prevent the formation of the biofilm. But if biofilm is formed and microbes start to kind of overcome the immune response on antibiotics, and it can happen, it causes inflammation in the joints and pain, whatnot. Okay, and in some patients who actually have biofilms formed on the surface of the joint, the entire joint has to be replaced again. So it's a problem. Another problem is catheters. Biofilms form on the surface of the catheter. Microbes can migrate through the from the urinary bladder up through the ureters into the kidneys. It's another problem. How to trace it, okay? How to detect it. So it's all biofilm formation is the big problem, and cleaning agents, now imagine this, things like vaporized hydrogen peroxide can kill biofilms, but it's crazy active and toxic and stuff. So it's a problem, okay? In some microbes, you can find so-called S layers, which function we don't really understand. It's a mixture of carbohydrates and glycoproteins, and it maybe just strengthens the wall, or may serve as the part, or may serve as the wall itself. You can see on this staining specimen the whitish area that surround cells. That's the capsule. Okay? Appendages. So we're going to talk about three of them: flagella, fimbriae, and pili. I bet you've heard about flagella before. The one and only purpose of flagella is what? Movement, yes. So this picture can scare the living daylights out of anyone, okay? But I'm not going to ask you about the fine details of the mechanism, how flagella rotates, okay? Pretty much you have a motor, a hook, and the filament. So if you imagine that my hands are the motor, this is the hook, and this is the filament, this is how it works, okay? Just like a propeller. Structure is slightly different between gram-positives and gram-negatives because of the different cell wall structure. What is interesting is the actual how motor works. Not in very much details, but remember in oxidative respiration, when we're going to talk about oxidative respiration more, but remember ATP is synthesized when hydrogen ions go through the molecule of ATP synthase. It's a gradient of hydrogen. Hydrogens diffuse through the large ATP synthase, and energy of that diffusion allows ATP to be produced. Now in this case, ATP is used to pump hydrogen, create the gradient, and when hydrogen diffuses through that motor, that energy of the diffusion, because this is your diffusion, energy of sliding down the concentration gradient, fuels up the flagellar movement. Does that make sense? Now, flagella can be arranged in different way. Monotrichous is on one pole. Amphitrichous on two poles. Mono is one. Amphi is both amphiphilic, amphitrichous. Lophotrichous is a bunch on one end of the microbe. And peritrichous, flagella all over the microbes, peri, around. Trichous, trick. Any idea what T-R-I-C-H means in Latin? Hair, yes, hair. Okay. Now, fimbriae. We don't really see them. Well, maybe you can count this. It's little hairs, hair-like tiny projections on the surface of some bacteria. Fimbriae help in attachment. So this is another example of virulence factor. Make sense? Because if microbe has fimbriae, it attaches better to the surface of, say, a cell and can cause infection with a higher probability. Make sense? Flagellum, can it be a virulence factor? Sure. Bacteria that can move has advantages, right? Pili. So these are pili. They long protein made projections, like tubes. And pili can form bridges between bacteria. They can form a line of communication between two bacterial cells. We call these structures sex pili. what essentially is sex? What is the purpose of sex? Right, but why not just dividing? Why people, well, because it's fun, but bacteria use binary fission. They divide cells, okay? So what is evolutionary advantage of sexual reproduction? Good. Exchange DNA, increase genetic diversity. Bacteria don't exchange DNA, or do they? Turns out, pili allow cells to exchange DNA. Now, if one cell has a trait, a fragment, a chromosomal fragment, or the other, let's say chromosomal fragment, gene that encodes the resistance to antibiotics, by chance, this gene can be exchanged with another microbe that does not have this trait. Does that make sense? So now both of them have this trait. So this gene can spread through the population. And the only thing that will determine whether it's going to spread or not is evolutionary fitness. Right? So if there is no antibiotics in the environment, then resistance to it, it's not advantageous. But if you start dumping antibiotics in the bacterial environment, then the ones resistant to it will survive better, and the entire population will acquire resistance. It turns out not only microbes of the same species, but sometimes microbes of different species or even genera may exchange genes, like Klebsiella and Escherichia coli. They can sometimes exchange genes, and that leads to the resistance spreading through various populations. Does that make sense? It's really scary. Finally, how microbes move using flagellum. Do they just, like, I'm going to walk there, I'm going to walk here? Well, they have a direction. This direction is determined by chemicals. So movement towards an increasing concentration of chemical attractant is called chemotaxis. For instance, a nutrient. Okay? Does that make sense? Microbes, it's like people go where the free food is. Put the announcement that there's pizza in breakers, and you will see people just walking towards breakers. So it's chemoattractant. It doesn't have to be chemical. Some microbes swim towards the magnet, magnetotaxis. We don't know why. Really, we have no freaking clue why they do it. Some microbes, probably photosynthetic ones, swim towards light. So it's phototaxis. That makes sense? So there are two steps in movement. One is called run step. When flagella rotate counterclockwise and microbe swims to wherever. And then tumble. When flagella start to rotate clockwise and microbe apparently moves around in a chaotic way. It turns out during the tumble step, microbe orients itself. Does that make sense? It orients itself according to the gradient of chemicals or magnetic lines, or light, right? So without chemoattractant, microbial move will be like this. With the chemoattractant, they don't swim directly, but it's going to be more like this way, okay? So you can see the general direction. Make sense? Any questions before we move on to the next step? Not really? Okay.